So can you see the live button now, Krishna?
Yeah, it is.
The live is on. Yes.
A few participants in the waiting room. So
Yes. So I'll admit that.
yeah, I'll start at 4:30.
Krishna.
Okay. Uh, Good evening, good morning, and good afternoon to our viewers from all over the world. Uh, Welcome back. We continue with our lecture series. Now we have the Shakuntala Devi session on computational biology. We had a session in the morning, and now we are going to have the second lecture of this series. Shakuntala Devi, uh, our India's beloved mental calculator, was indeed a woman who has put mathematics on a world forum like no other. Having amazing mental mathematics abilities, which was discovered at a very young age, she has amazed the world with the speed and accuracy of her calculations all throughout her work. Famously, when her abilities were tested at the University of California, Berkeley, she indeed amazed the researchers by solving impossible sums such as the seventh root of a 9-digit number or the cube root of an 8-digit number even before someone could write it down. In 1977, she famously solved the 23rd root of a 201-digit number in 50 seconds, beating the speed of a UNIVAC 1101 computer, a state-of-the-art computer at that time, which earned her the nickname human computer. Apart from that, she has also authored several books and also published the first ever study on the society's understanding of homosexuality in India, a work which is considered pioneering in this field. So, in honor of India's beloved mental calculator, the human computer Shakuntala Devi, biologically speaking, is proud to present the Shakuntala Devi session on computational biology. We had a talk in the morning featuring Dr. Lipi Thural of CSR IGIP, who talked to us about her lab's work, as well as the progress and the need of learning computational biology or basic computation for any biologist. Now we have with us Dr. Dr. Tripathi is a computational biologist and currently a senior principal scientist in the Structural Biology and Bioinformatics Division at CSR Indian Institute of Chemical Biology, Kolkata. After completing her PhD in plant molecular biology the Center for Plant Molecular Biology, Hyderabad. Uh, she joined DSQ Biotech Limited in Chennai as a scientist before she moved to Avestagen Grain Private Limited in Bangalore, where she served as the head of the Bioinformatics division. She then joined Virginia Bioinformatics Institute, Virginia Tech, USA, as a senior research scientist in 2002, before returning to India as a Ramalingoswamy Fellow to join ISAB in 2012, where she has served till date. She serves as a research advisory committee member at the National T Research Foundation in India. She serves as a guest associate editor in Frontiers in Genetics for Evolutionary and Genomic Microbiology, while also being a senior editor of MPMI, Molecular Plant Microbial Interaction, an American Phytopathological Society journal. Her primary interests are computational biology, computational genomics, and bioengineering. Her lab works on the genomics of indigenous microbes such as cyanobacteria using second and third generation sequencing techniques to decode their genomes. They analyze transcriptional changes occurring in microbes under various stress conditions with the goal of engineering these genes for the commercial production of biometabolites and active compounds. With that, we welcome you, ma'am.
Many thanks, Krishnamurti, and also the organizers of Biologically Speaking. I'm really happy and proud to see that you guys are taking so much pain and doing this, organizing this lecture. And and I think you have also tweeted and many people have joined in. So I have started sharing. So let me know if you can see my slide or
Not yet.
not yet. Okay. Let me try sharing my screen.
Yeah, right. Yeah, you can see it.
So today I will talk about, although you have classified all your sessions into different prominent scientists such as Shakuntala Reddy, and I'm really very glad that you have included computational biology. Although I'm not going to talk deeper about computational biology, but I will tell a story that is emerging out of our biocomputation, which involves genomics. And if at some point of time you cannot hear me, just stop me and ask me, okay? So I'm just
Yes,
assuming
ma'am,
that you
sure.
all can hear me and see my slides. So today I'm going to talk about a small part of the work that we did in collaboration with Calcutta University and with the help of computational genomics and the wonderful insights that we got into this amazing world of fungi and endophytes. So what are endophytes? As we know that endo means inside and phyte means plants. In nature, we see so many things. We see plants, animals, and that's all we can see. But there are a lot of microbes that we cannot see, and they do a lot of work. So this particular job, this particular work was mostly done by these three amazing people here. I'm showing photograph. Here is Professor Anindita Seal. She's a professor in biotechnology from Calcutta University. She is the one who actually initiated all this project that I'm going to present today. And the next person is Dr. Dia Sen. She was a pool scientist in my lab. Now she's in New Zealand working as a scientist. And this is Dr. Cornelia Paul. She is a graduate student of Dr. Anindita Sen. So all this thing began when Anindita was very curious and she went to a place in Calcutta, in West Bengal called as Jaduguda, which is known for having uranium contamination. And as you can see here, this water is actually heavily contaminated. And what is expected out of this is you would see that you would hope that there is nothing growing there. But to her surprise, she saw that lots of healthy plants are growing, and these are actually monopods. Monopods are a different category of plants which has only single cotyledon. So she was out of curiosity. She has taken some samples from here and she took it to her lab. And since this plant is called as typha, which is a grass, and she thought that if, and she started growing this organism, which is a yeast. Yeast is like a fungi, she grew in the lab, and she started doing some experiments, and she injected this to another monopods, that is our staple food, which is rice. And she found that when she has injected this to rice plant, she saw that there is amazing growth. As you can see, between control and treated, the plants are much healthier and they have more vegetative and reproductive growth. And this is a microscopic picture which shows that this yeast is actually an endophyte that is staying inside the cells of the host plant. And this is another picture from the root which shows the endophyte is there. And another thing they have noticed was, as you know, that in case of uranium mines or any other places which are very nutritionally depleted areas, where the fundamental nutritional components such as phosphates and nitrates are not available to the plants, and phosphates are not usually soluble by the plants. Plants cannot solubilize those and cannot uptake them very readily. So she had a hunch that maybe this fungi, which is named as Rhodotorula mucilaginosa, and it has a strain name which is named after the Jaduguda mine, JGTA. They did some experiment in a media in which they grew the plant, grew the fungi, and you can see the halo around this, which indicates that there are this fungi is secreting certain enzymes which is solubilizing the phosphate. And that is also reflected in the experiments, where in the control plant, when it is grown in a soil which is depleted of phosphate, in 10 days they all die and they become chlorotic and they do not have enough growth. But the ones in the same soil where they are inoculated with rhodotorula, you can see that there is a huge difference in the uh, health of the plant. The plant is very green and the root growth is very high. So, in order to understand this, so there the story begins. So they came to us, and we decided that let's study the genomics of Rhodotorula mucilaginosa, and we will also study the transcriptomics, the interaction using computational biology. And we did a lot of series of experiments. We did a series of microarray experiments, which is to know that whether which transcripts are up and down regulated when Rhodotorula was injected into the rice plants. And it was found that most of the when the enrichment analysis was done, most of the auxin related growth factors, that is the growth factors for the rice plants, were upregulated. And also the genes which are responsible for ammonium, for nitrate assimilation and nitrate fixation, and nitrogen permease proteins were also highly upregulated. And there are several other proteins like lyse M, and which I'm going to talk about, which is really, really very interesting story, were also upregulated. So this is the beginning of the finding, and this was getting more and more interesting. And this story was actually, this is also published, but I'm just taking you back into how the whole story unfolded. And just to show you what happens in nature, we all know that nitrogen is a very important element in every living being, and more so for the primary producers. The primary producers are the plants which fix or which get the nitrogen from different sources, and they incorporate into amino acids which are consumed in turn by animals or humans and other people. So that is how we get our nitrogen, mostly is coming from the primary producer. But you will be surprised to know that the plants do not have a mechanism in which they can utilize the atmospheric nitrogen. They can, however, they can take the nitrate which is present in the soil by nitrate transferase gene, which is coming into the roots of the plant. And then there is another enzyme which is called as nitrate reductase, which reduces it to nitrite. And this nitrite is further reduced by nitrate reductase, which is converted into ammonium. And this ammonium is taken in by this glutamate pathway, where it is again distributed into production of different amino acids and subsequently into proteins and other nucleotides. So this is a fundamental aspect of how the nitrogen is taken in by any primary producer or plants. So there comes another species of organisms which are prokaryotes, mostly cyanobacteria, they do biological nitrogen fixation. That means that they utilize the atmospheric nitrogen and they convert it into ammonium, which is given back readily to the higher plants, which can be taken in and they can start utilizing it. So this is, but on the contrary, we found that when JGTA was injected into higher plants, there are lots of genes which were, as I said earlier, that many genes which are encoding nitrate assimilation pathway and nitrate transporters, and all the genes which are responsible for amino acid synthesis were actually upregulated, and nitrate and ammonium transporter genes were also upregulated. So that was a kind of starting point which made us think into what is happening inside the inside this organism. So in order to understand this, we started with the genomics, which is what my lab mostly do. We focus on the genomics, we understand, we sequence the genomes and we try to decipher them. So we took the samples from Calcutta University and we sequenced the genome of this organism using second and third generation sequencers. The first one is we have generated 74,892 reads using Oxford nanopore sequencing technology, which is about 42x coverage of the genome. And also we did supplementary sequencing by Illumina, which is quite higher depth of coverage, which is 262x coverage. And we combine both of them and we use different assembly methods. So the reads are nothing but the sequencing is producing short fragments of reads which are not usable directly. What needs to be done is they need to be uh, aligned together so that we find bigger chunks, and those bigger chunks can be used for analysis to find what is actually there encoded in the genomes of this organism. So we have used several methods, and one of the methods is to just estimate the genome size of this organism, and we used one program called as gamer count extract. And with that, we found that this genome size is about 21 MB. And using different assembly methods, we have generated different types of assemblies, and out of which we picked only two, which is A1 and A2. And A1 assembly contained 341 scaffolds. That means 341 fragments contained the whole genome, whereas A2 had only 46. So in genome assembly, you'll see that the lesser number of scaffolds you have or the longer scaffolds you have, the better the assembly is. So from A1, when we discarded all the short assemblies, which is less than 2K, we came up with A2. And by doing A1 and A2, we from A1 to A2, we did not actually lose a lot of genome. They were almost of the similar size, but on the other hand, we could see that it is more contiguous and it is having bigger contig sizes. So by N50, the larger the N50, the better is the genome assembly. So N50 means that half of the genome is contained by scaffolds which are at least this much of size. That means if you have half of the assembly, if the larger you have, that means that you have more contiguous fragments. So we chose A2 for all our subsequent analysis. Then we started looking at the genome, and then we found that it is actually having a lot of fascinating insights. And we picked up another 5 organisms of similar genome genus from the GenBank and from other sources. And one such organism was C2.51, which was isolated from actually very cold region, Antarctica. And in fact, they could not assemble it. It was very fragmented. So we took it out and we had our in-house algorithms in which we have reassembled those, and we try to align these two organisms, two assemblies, and as you can see that the genome and the proteome, they are aligning perfectly. And this is also another map view to show that they are agreeing with each other. In addition to that, we also found a mitochondrial genome of this organism for the first time. Then we curated the other three species that we got from the Gen Bank, and we found that our organism is actually very close to the Antarctica organism, where they are grouped together, and they share a lot of genes. Like they have about 4,315 core genes, which are common between all of them. This is very very meaningful. This means that even though these organisms have been separated physically for a very very long time, still they share a lot of core genes, which is responsible for maintaining its endophytic nature, biological nature. And the next thing we did was we compared them because, as I said, that Rhodotorula is a yeast. Yeast belongs to a class of fungi called as Basidiomycetes. And Basidiomycetes is a huge class where you will have these mushrooms, also Basidiomycetes, and there are also many other fungi which are parasites and saprophytes. So, what is a parasite? Parasite is an organism, fungus, which attacks the plants and kills it, whereas endophytes are the organisms which attack the plant, but they have some kind of mutualistic relationship. That is, the host is also benefited, and the endophyte is also benefited. They do not kill the plant. So here, what we tried to do was we tried to compare them with other Basidiomycetes fungi, which have different characteristics. Like some of them are parasitic. For example, Ostilagomyces is a parasite on wheat plant, and you can see that they share a lot of genes, especially some of the genes which are responsible for initiating and host infection, and they have about 25 genes which are common between these two. So you may ask that why Rhodotorula, which is a growth enhancing organism or an endophyte, should have any gene which is responsible for carrying out infection. So in order to understand this, first of all, when there is an endophyte, the endophyte has to get into the host plant. So how does the endophyte get into the host plant? In order to get into the host plant, it has to first enter through the cells. And when the something is trying to enter into a cell, the first defense mechanism of a plant is to try and recognize it and to try and destroy. So plants have several defense mechanisms. One of them is a production of salicylic acid. Salicylic acid is produced by the plants and secreted to the environment. And that salicylic acid actually kills the microbes. So endophytes such as Rhodotorula, we found from the genome that they have a salicylate hydrolase gene, which degrades the salicylic acid, which is produced by the host plant. And also another interesting thing we found was they have a class of proteins called as chitin deacetylases. Chitin, as you know, is the cell wall composition of fungal cell wall is partly cellulose and chitin. And chitin is recognized by the host plants. So once the host knows that there is a chitin nearby, it triggers a defense response. So in order to stop the plant or the host from triggering a defense response, what Rhodotorula has done is they have a set of enzymes which actually converts the chitin they have on the cell wall into chitinase, which cannot be recognized by the plant receptors. So that way they evade the detection of the host surveillance system. And also they have another class of proteins that we have recognized from the genomes, that it has undergone expansion. What does it mean by expansion? The expansion means that when you see a genome, you'll find that some of the genes, they are not only present in single copy, but they're present in multiple copies. So what happens when one organism wants to increase its fitness, they try to create more copies of the gene, and they expand that particular family of genes. So we have particularly found that in case of Rhodotorula, it has an expansion of this particular family of genes, that is proteophosphoglycans, which are responsible for cell invasion. So initially an endophyte has to establish a response or establish a infection process where it has to get into the host plant uh, by evading the host surveillance system. And those sets of genes were found very much intact in this genome of this organism. And another interesting feature we found is when we compared this organism with other pathogens, we found that the pathogens are carrying a genomic cluster, which is called as 19A cluster. And this 19A cluster codes for genes which are effectors. The effectors are the proteins responsible for creating for triggering a defense mechanism in the host plant, which can be recognized by the pathogen, and in turn that disables that defense mechanism. So in other words, it causes a um, it causes an attack on the host plant. So this locus has been found to be deleted in Rhodotorula. So that may be one mechanism by which it has been adapted from being a pathogen to a endophyte. And also there is another gene, another class of proteins which is called as NMRA. And NMRA is a transcriptional regulator. So what does it do? As we know that when there is a plant or there is a pathogen or there is some organism, they know that where the nitrogen is coming from. But in case of an endophyte, what it does is it has a very clever mechanism. It knows that my nitrogen is going to come from either nitrate or from some other source. So it has a switch. And that switch is the NMRA, which switches between different nitrogen assimilation pathway or nitrogen metabolism depending on the nitrogen source available. So this is actually the trigger for us to understand what exactly is happening inside the cell. And this is a very, very important point that we have found out from the genomic studies. So I'll come to that story a little bit later, but let me also impress upon you what we have found with this particular organism. So Rhodotorula also, although it is adapted to India, as I said, we had found another close homologue which was found in Antarctica. That led us to think that they may be having some genes which are adaptive for cold tolerance. And we found indeed there are 5 genes which are giving resistance to cold, which is giving for cold adaptation. So when we grew these plants with Rhodotorula and yeast, as you can see, just common yeast, they grow well in 30 degrees centigrade. But when we reduce the temperature to 5 degrees centigrade, we found that the yeast doesn't grow, whereas Rhodotorula happily grows. So that means the cold tolerant genes are quite active in this organism. And in addition to that, there are also carbohydrate binding modules which were found in this organism. And these carbohydrate binding modules are similar to necrotrophs and hemibiotrophs. What are necrotrophs? Necrotrophs are the organism where the pathogen is causing an infection, but it turns into a saprophyte. That is, it lets the tissue or it lets the host plant carry out programmed cell death such that there are dead tissue and this organism feeds upon the dead tissue. That is called as necrotrophism. And hemibiotrophism is where the infection first occurs, and at that time the pathogen doesn't want the host to have programmed cell death. It wants the host to be alive so that it utilizes some of its cellular mechanism for its growth to a certain extent, and after that it encourages programmed cell death and it carries on uh, necrotrophism inside the plant. Whereas biotrophism is something where the pathogen doesn't want the host to be killed at all. So it wants the host to be alive so that it utilizes everything inside the host. So here we found that the characteristic of Rhodotorula was similar to more or less to necrotrophs and hemibiotrophs rather than the biotrophs. And this is a casein profile where we have studied all the carbohydrate degrading or carbohydrate metabolizing enzymes using DPCAN database, and we have compared with all other 9 other species, and we found that exactly how many genes are common between them. And this also gave us a lot of insight into what kind of carbohydrate metabolism is taking place inside this organism. And another interesting gene we found here was a gene which is called as isopentylyl pyrophosphate isomerase, that is called as IPP isomerase. And we all know that plants need a phytohormone called as kinetin for its growth and most of the metabolic processes. So this kinetin occurs inside the plant in a very inactive form until these enzymes this enzyme acts upon it and makes it active. So this enzyme was found with rhodotorula. So in other words, if this is present, if there is an endophyte inside the plant, it actually makes the plant succeed in its growth and its metabolic processes. And this is also very this led us to believe that how a yeast, which is a single cellular organism, uh, can harbor something so important. And accidentally also it was discovered that this organism is existing in two forms. That is, one is this unicellular form like yeast form, and the other one is a filamentous form, which was quite evident from microscopic studies. And from our genomic study, we also found this was not known in yeast-like fungi or in Rhodotorula. It was never documented before that it contains a mating loci. A mating loci is responsible when there is a sexual reproduction taking place inside the fungi, which was not known to be there because it's a yeast-like organism, and it was believed that it was always propagating by means of budding. So since we found this mating loci, this led us to believe that this may be having a secret sexual stage inside the organism. And also we found very interesting in this organism was, as I showed you earlier, that this is the nitrogen assimilation pathway. Whereas from our genomic study, we found that there are two genes, that is nitrate transporters and nitrate reductase. Those are missing in all Rhodotorula species. That means that these organisms are incapable of assimilating nitrogen. So this is a very interesting thing because we know that when the host is growing without nitrogen, it is growing very well, and Rhodotorula itself can grow very well without nitrogen. And when it is having a non-functional or defunct nitrate assimilatory pathway. So this led us to the third question: is what is actually happening inside this organism? And this is also some accessory slides to show you that when we also believe that this Rhodotorula is actually not acting upon by itself. It is having a third organism, which is, this is a significant discovery, and this is what was published in Plant Cell last year, where we showed that there is existence of another organism which in turn helps Rhodotorula to fix nitrogen because it is incapable of nitrogen assimilation, and it is also helping the host plant for this. So here in this case what happened was Anindita and her group, they try to uh, treat this JGTA into with different antibiotics in order to cure it of any bacteria, and they have found another a strain of that Rhodotorula, which was completely cured of bacteria. Although there were some bacteria, but it was almost like it was non-existent. So when they grew them in absence of nitrogen, as you can see that JGTA works very well when there is no nitrogen in the media, but this strain 11, which is not having any bacteria, has no growth. So this again led us to believe that there is another bacteria which is actually helping this yeast to fix nitrogen, and this nitrogen in turn is giving back to the host plant, and there is actually a tripartite interaction. So here then we had to again go back to our genome and to go back to the reads, and we scratched our heads and we tried to find out. So what we did was we took out all the raw reads and mapped back to all the available genomes, including some of the nitrogen fixing genomes in NCBI. And we found that there were about 1.85% of the reads which were not assembled into anything. And those reads mapped into a nitrogen fixing bacteria, which is Pseudomonas sturgeonii. Although the number was not very high, but still we could find some of them. And using that, and then we had a hypothesis because we believed that in the filamentous form of Rhodotorula, the bacteria is associating with this, whereas in the yeast form, the bacteria doesn't. So this microscopic evidence also tell us that Rhodotorula in fact has a bacterial association when it is in a filamentous form. So this is the hypothesis we proposed, which says that when a rhodotorula is in a filamentous form, uh, it is in it is reproducing sexually, and then it comes in contact with the bacteria, which gets into the mycelium, and this in turn gets into the host plant, and then it helps the host plant, then it uh, helps the host plant to do growth enhancement, and the cycle is repeated. So this led us to several insights from this genomic, which is one interesting characteristic, that is discovery of a sexual phase that was using a bacterial association which was not known before, and incomplete nitrogen assimilation compensated by a tripartite interaction, and host evading mechanism by modification of chitin, and plant growth regulators supplied by endophytes. So this was really, really interesting. And in the end, I just would like to say all of you thanks a lot for giving your time and listening to us. And this is my group. This is my lab family, without which I cannot, I won't be here in the first place.
Thank you, ma'am.
So I can take questions if you have.
Yes, okay. The first question is from Shonchari Gorai. What's to do with if rhodotorula cannot fix nitrogen, can they use fixed nitrogen by the associate flora? Is this association symbiotic?
Yes, the bacterial association is symbiotic because the bacteria fixes nitrogen, and it helps rhodotorula, and of course it is symbiotic.
Okay. In extension, I was wondering if the bacteria itself is a nitrogen fixing bacteria, why does it need rhodotorula as an intermediate with the plant?
It is actually very interesting. In nature you will see that why a fungi also needs a plant. Fungi doesn't need a plant. If you look at the genomes of this organism which are associating with a host or anything else, there is a genome reduction. What they do by that is they get rid of some part of the genome, and they are complemented by the host plant. So this is a unique relationship in which that they give and take. So similarly, a bacteria which may be taking something from the host, which is a fungi here. So that is also a metabolic fitness for the bacteria. The bacteria doesn't have to harbor all the genes that it needs. So it gives the nitrogen. In turn, it takes something else, maybe the minerals. So most importantly, all the fungi are known to produce like phosphate and other minerals. They supply minerals and to the higher plants or other organisms. That is how they have a very good relationship, and they exist in nature all the time. They occur together.
Dr. Adumugam Madhumadar wants to know how many cold adaptive genes you have identified and how many are essential.
There are 5 cold adaptive genes we found, and out of which we found 2 were active, and then others were just silent. Maybe they were compensating for if there is something happens to these genes, they become non-functional, then they'll start expressing themselves.
Uh, in continuation with this, uh, it seems that despite being separated for so many million years,
Mhm.
why has the Indian variant possibly retained the cold adaptive genes? I mean, theoretically, it's not much required in Indian conditions, but have lost it evolutionary. Can you elaborate on that?
Yeah, actually, the distance or the separation is how many years old we cannot tell at this point of time. We have not analyzed to know that how many years they have been diverse from each other. But if you can see the genomes are actually quite similar to each other. The C251 and this one. I think the separation was not very ancient, probably.
Uh, there's one more question, if you are okay with it. Bina wants to know in your genome sequence, shouldn't the bacterial genome be also contained? I
Yeah, that
suppose
that
you
that is
yeah
what
you described.
exactly we found out. That's what I also said that in my
Yeah.
presentation. That when we assemble those, we assemble. So when you assemble a genome which contains different types of reads, they've been into different assemblies, right? So the fungal genome was separated, and some of the reeds which are not found in the fungal genome, as I said, 1 point some percent of the reeds, they contain the bacterial genome, bacterial reeds.
Okay.
And that is how
Thank you,
we
ma'am.
discover. Okay.
Thank you so much. It was a wonderful lecture.
Thank you, guys. Okay, bye-bye.
With this, we come to the conclusion of the Shakuntala Devi session on computational biology. We'll next go forward for our next lecture, and this will be taken over by Sundaram Acharya. Sundaram is a research fellow at CSR IGIB in India. So, if you're there, it's over to you.
Yes, Krishna. Yeah. Am I audible?
Yeah.
Right. A warm welcome to the session after the name of Asima Chatterjee. So this session is on rising stars in biology, which had been named after Dr. Asima Chatterjee, which holds the distinction of being the first woman to earn a doctorate in science from an Indian university. Dr. Asima Chatterjee was born in 1917 in West Bengal, India. Growing up in a middle-class family in Calcutta, she was encouraged to pursue academics. She completed her graduation in chemistry honors in 1936 from the Scottish Church College of the University of Calcutta. She went on to complete a master's degree and a doctorate in organic chemistry in 1938 and 1944, respectively, from Raja Bazar Science College, University of Calcutta. In a scientific career spanning over 40 years, she conducted research on various alkaloid compounds and natural products chemistry. She discovered compounds with anti-epileptic activity in Marsilea minuta and anti-malarial compounds in Picorrhiza, Kuroa, Alstonia scholaris, and so on and so forth. Her research led to the development of an epilepsy drug named Ayush 56 and several anti-malarial drugs. She published around 400 papers and has been a recipient of numerous prestigious awards, including C.V. Raman Award, P.C. Ray Award, and the highly coveted S.S. Bhatnagar Award. She left us in 2006. She was a woman who inspired millions of aspiring scientists to dream and achieve their dreams with hard work and dedication. Honoring the first female doctorate of India, Biologically Speaking is proud to present the Dr. Asima Chatterjee session featuring rising stars in biology. With this, so in this session, we have two speakers. The first is Parej Nath, Institute of Life Sciences at Bhubaneswar. And the second speaker in this session is Dr. Sweta Chakrabarti, currently based in IISc Bengaluru. With this, I'd request Parej to join us. So Parej is currently a doctoral candidate who is currently based in Institute of Life Sciences, Bhubaneswar, India. She is currently at Chauhan Lab under the supervision of Dr. Santosh Chauhan. Recently, she had published an article titled IRGM Links Autoimmunity to Autophagy as a first author in the prestigious journal Autophagy. She has been awarded the highly coveted Sun Pharma Science Scholar Award in 2020. With this, I'd like Parej to join us, and the stage is yours.
Thank you. Thank you. Am I audible, Sundaram?
Yes, you are loud,
Okay.
clear and loud.
Okay. So I'll share my screen.
Yes, please.
So my screen is visible, I guess.
Not yet.
Okay. Let me just wait. Is it visible now?
No, not yet. Have you shared?
Yeah, I have done this share screen.
Mm-hmm.
And is it now?
Yes, now it, yeah, now it's coming. Yeah.
Okay.
Go to
Okay,
full
now.
screen. Yeah.
Yep. Okay. Yeah. Okay.
Yes, now it's fine.
So thank you. First of all, I would like to thank the organizers for giving me this opportunity to come here and present my research work, which I've been doing in my PhD. So first of all, let me introduce myself. I am Paryaj Nath, a final year grad student working with Dr. Santosh Chauhan in Institute of Life Sciences, Bhubaneswar. And today I'll be presenting a part of my PhD thesis entitled Understanding the Role of IRGM in Innate Immune Homeostasis. So we all know that our body is under constant threat from different kind of pathogens, be it bacteria, viruses, any kind of parasites, and our immune system is playing its part very well in protecting us from those kind of attacks. Now, innate immunity can be called as the first line of defense against any such kind of pathogenic attacks, and it does so by generating response as a result of by producing different kind of biological and chemical compounds known as cytokines, which intensifies the response further. Now, one such response is called inflammation. Now, inflammation is of two types, acute and chronic. Now, chronic and persistent inflammation can result to various kind of diseases like autoimmune disorders, neurological diseases, arthritis, and even cancer. So it is very important to have a balance between required amount of inflammation and excessive inflammation so that our body does not go into shock. Now, talking about and coming back to cytokines, type 1 interferon is a kind of cytokine which is generally secreted upon viral infection. Now, from type 1 interferon today, now I will come to my protein of interest, which belongs to this interferon inducible GTPase superfamily and comes under immunity related GTPases subfamily or IRGs. Now, IRGs again have different groupings like IRG A, B, C, so on and so forth. But my protein of interest is IRGM. Now, why IRGM and why not any other IRG like IRG A or B? That is because of its very interesting evolutionary history. This gene, IRGM, was lost some 50 million years ago in old world monkeys. But then again, with series of mutation, it was again brought back to life in primates, primarily gorilla, chimpanzees, and humans. Although in a truncated form, but it was functional. Now, rodents have 23 IRGs, which are functional, but humans, we only have one IRG, and that is IRGM. Now, a protein which was dead, but then again was brought back to life, must be having some very important role in our body. And indeed, it has. Literature studies have shown that polymorphism in this gene IRGM is linked to several kind of inflammatory disorders like Crohn's disease, atherosclerosis, multiple sclerosis, gastric cancer, and in all of these diseases, IRGM plays a protective role. IRGM is also a very important autophagy protein. It is one of the sentinel player in the core autophagy machinery. Besides, IgM also plays protective role against bacterial diseases like Mycobacterium tuberculosis, Salmonella infection, and Listeria monocytogenes. But till date, the molecular mechanism by which IgM is playing this protective role against inflammation and inflammatory diseases, it's still elusive, it's not known. So recently, our lab, in a study, we have just shown that IgM is acting as a negative regulator of inflammation by impeding the polymerization and formation of NLRP3 inflammasome, both in human as well as in mice cells in perspective to Crohn's disease. So keeping this findings in our mind and also the literature whichever we have, we were curious to know that in a broader spectrum, how IgM is regulating the cellular inflammation. We were very curious to know. So we started with transcriptome analysis. We wanted to know what will be the difference in the transcriptome of the cells which express IgM and the cells which do not have IgM. So we have taken control and IgM knockdown HT29 human cells. These are colon epithelial cell lines, and wild type and IgM knockout mice brains. And we found that the major pathways that were getting upregulated in the absence of IgM belong to the immune system or the cytokine signaling pathways. And most of the genes they fell under the interferon signaling or type 1 interferon signaling pathways. So from this RNA-seq, we next wanted to validate this data. So we have chosen some of the sentinel genes that belong to the type 1 interferon pathways like RIGI, OS1, the STATs, ISG15, MX1, and 2. And we found that at the RNA level, both in the human cell as well as in the mice brain, these genes were significantly upregulated. Now, after this, we came to know that in the transcription level, the genes are getting regulated. But what happens at the protein level? Now, this is a schematics that I have given. This is a type 1 interferon signaling pathway, which begins with the cytosolic sensors. These are the PRRs, pathogen recognition receptors, RNA and DNA sensors respectively. Then comes their adapters, the kinases, the transcription factors. Then they go by paracrine signaling. The interferon alpha and beta comes and binds to the IFNR receptors. Again, by means of the cycle of transcription factors, the ISGs are produced. Now we started to check the status of each of these players at the protein level, and we were like very surprised to find that starting from the RNA sensors, RIGI, MDA5, TLR3, IFI16, CGAS, as it's very clear from this Western blot data, they were all upregulated. Then we came to the adapters, as we can see, it's upregulated. Then the phosphorylated form of the kinases, TBK1, the phosphorylated form of the transcription factors, IRF3, IRF7, and STATs, they were considerably upregulated till the extreme end ISG, that is MX1. So from this, we have hypothesized that when IgM is not there in the cells, the PRR signaling pathway, the pathogen recognition receptor pathway, they are in a constitutively activated state. Now, it's already known that IgM degrades some of the PRRs like NODs by autophagy to impede bacterial signaling inside the cells. So we were curious to know that whether in this case, in case of type 1 interferon signaling also, IgM does have any kind of role like that. So we, by series of immunoprecipitation experiments, both at the endogenous level as well as in the exogenous level, we found that IgM specifically interacts with RIGI, CGAS, and TLR3. This is an endogenous system, and here we have co-expressed IgM with their respective PRRs to find that yes, they're interacting. This is an in vitro experiment where we have done the GST pull down assay to find the similar result. Now, what is the next step? It is interacting. Does it degrade the PRRs? Yes. In both stably IgM expressing HT29 cells as well as in exogenous systems where we have overexpressed IgM with the PRRs, we can see that it's very clear that IgM is degrading the specific PRRs, RIGI, CGAS, and TLR3. This luciferase assay data also tell us the same story by that when we are expressing IgM with this PRRs, the ISG, the downstream ISG response is getting reduced as compared to when IgM is not there. Now, till far we know that yes, IRGM is interacting with the PRRs and they are degrading. It is degrading it. But then, what is the mode of action? Does it use proteasomal degradation machinery pathway or autophagy machinery pathway to degrade it? So we used two of the chemical blockers of autophagy, bifilomycin and chloroquine, to find that when autophagy is blocked in these cells, the degradation of these PRRs, Rigai, Cgas, and TLR3 are getting restored. Similar result was found when we blocked autophagy by genetically knocking down autophagy genes like Beclin1 and ATG5. These two are very important autophagy genes. When we are blocking down autophagy by genetically knocking down these two genes, the degradation of the PRRs are getting restored, which is very clear from this Western blot data. Now, when we talk about autophagic receptors or adapters come to our mind. So we were curious that whether in this entire process, any kind of autophagic receptors is involved or not. So we have taken or we have chosen some of the very well-known autophagic receptors like P62, NDP52, NBR1 and found a very strong interaction between IRGM and P62. So to further confirm the role of P62 in the degradation of this PRR via IRGM, we did this series of immunoprecipitation assays where we found that the interaction between IRGM and the respective PRRs were getting reduced when P62 was not there in the cell. Not only this, we also found by immunoprecipitation that the PRRs, IRGM, as well as P62, they remain in a complex. So this part, the story is like in the presence of IRGM, it recruits P62 to bring C-gas and regide to the autophagic degradation and keeps the type 1 interferon response under check. Now, this far it was everything going fine, but then a very obvious question that came to our mind that when IRGM is not there in the cells, how come the sensors they are constantly on? Like they are always in an on stage, and why the interferon response is so high under basal condition, under steady state condition. So we went back to the literature and we came across studies which have shown that mitochondrial reactive oxygen species is one of the main reason for having heightened type 1 interferon response inside our body. So our next step was to check the condition of mitochondria and mitochondrial ROS inside the cells which don't have IRGM. And surprisingly enough, we found that cells lacking IRGM showed swollen mitochondria. These are wild type and IgM knockout mice BMDMs, and it's clear from the IF that the cells which don't have IRGM show swollen mitochondria, which is a hallmark of hyperpolarized and damaged mitochondria. Now, mitochondria gets accumulated in a case when mitophagy is defective. And surprisingly enough, we found elevated levels of two most important mitophagy proteins, parkin and pink1, when IRGM is absent from the cell. Parkin and pink also get accumulated when mitophagy is defective. Not only this, parkin and pink, they also co-localizes with the mitochondria, and this level of co-localization is way much more higher in the cells which lack IRGM as compared to the normal cells. Now from here, we further wanted to check or validate the health of mitochondria. So we did this a gold standard experiment. This is called a seahorse mitochondrial stress analysis system. It demarcates between healthy mitochondria and unhealthy mitochondria. So as expected, the cells which don't have IRGM showed unhealthy mitochondria as compared to the cells which have IRGM. Besides, we used these three dyes, CMX ROS, Mitosox red, and CellRox green. CMX ROS is used to measure mitochondrial polarization, membrane polarization. And as it's clear, in IRGM knockdown THP1 cells, CMX ROS staining is more, which means that the mitochondria are hyperpolarized, they are damaged. Mitosox depicts the amount of superoxide entities released by the cells, and CellRox is a measure of mitochondrial ROS, which is generated by the cells. And both these entities, by facts, we are showing that they are increased or heightened in cells which don't have IRGM. Now, mitochondrial or increased mitochondrial ROS production leads to genomic instability, mitochondrial instability, which leads to release of danger-associated molecular patterns in form of double-stranded DNA and double-stranded RNA from the cell, which gets sensed by the cytosolic sensors like CGAS, Regide, TLR3, not TLR3, CGAS and Regide. And results in the heightened type 1 interferon response. Now, these two IF images shown that in mice BMDM, when IgM is absent, both double-stranded DNA and double-stranded RNA, these are way much higher as compared to the cells which have. Plus, they are also co-localizing with their respective sensors, DNA with cGAS and double-stranded RNA with RIGI. Now, to further confirm that we know that these entities are higher in cells which are absent of IgM, but the entire response that we are speculating that in absence of IgM, type 1 interferon response is going high. Is it mitochondrial dependent? Like is the response coming from the mitochondria? So we did this very fancy experiment. We have created HT29 cells devoid of mitochondria. We treated the cells with ETBR, which results in the absence of mitochondria. These are called row 0 cells. And we found that normal IgM knockdown HT29 cells showed more type 1 interferon response as compared to cells which lacked both IgM as well as mitochondria. So from this experiment, we were like sure that yes, the heightened type 1 interferon response is because from the danger associated molecular patterns, the DAMs which are being released from the damaged mitochondria, which in turn might get sensed by either cGAS and RIGI. How did we prove that? We did double knockout of these receptors, cGAS and RIGI together, cGAS and RIGI with the respective adapters like STING and MAS, and separately we have double knocked down the transcription factors IRF3, IRF7, and STAT1, and we have checked the interferon response, the IgG response in mice as well as in humans. And we find that the entire response is going through cGAS, STING, and RIGI MAPS pathway. So, in a nutshell, the story this far is like when IgM is absent, mitophagy gets defective, which leads to the accumulation of damaged mitochondria inside the cells, which releases double-stranded DNA, RNA in form of DAMs. It's getting sensed by cGAS, RIGI, STING MAPS pathway, and which is letting the heightened type 1 interferon response in the cells. Now, and again, like I've mentioned in my one of my introductory slides, that type 1 interferon response is the major antiviral immunity that our body presents against any kind of viral infection. And from our RNA-seq data, also, we found that some of the major viral restriction factors like HER-C5, SAMHD1, I5TM3, which are involved in different stages of viral restrictions, they were significantly upregulated in the cells absent of IgM, both at the mRNA and the transcription level as well as in the protein level. So from here, we speculated that depletion of IgM takes the cells into an antiviral state. But in real time, does it actually happens like this antiviral state of the cell? Is it in actually able to restrict the viruses? So what we did, we've taken live viruses, chikungunya as well as Japanese encephalitis virus, and infected these cells, control and knock down IgM HT29 cells with these viruses, and we were like too much surprised to find that the cells which did not have higher GM, they highly restricted the viruses. This was in vitro condition, and we wanted to see that what happens when we take this or we infect the mice, like what happens in the in vivo condition. So we have taken wild type and knockout IRGM mice and injected chikungunya virus with them, and the result was like starking. The knockout mice, the mice that did not have IRGM, they were like they did not show any sign of paralysis or any sign of infection. Their vital stats were quite good, plus their survival rate was way much higher as compared to the wild type mice. So it's like the cells and like not us, but when IgM is not there, the cells are highly resistant to virus. Here I'm talking about chikungunya and JV, but besides this, we have also used West Nile virus, a Zika virus, and more recently we have started working with SARS-CoV-2 virus. And it's like it's very starking that even IgM depleted cells, they are restricting the SARS-CoV-2, which is like very surprising, and it will be I think a very good finding. So this work is under revision, like we are doing it. We have to do a lot, but I hope we'll be able to present something really good. So but the take home message is that when IgM is absent from our cells, it invokes a cycle of inflammation, and it is like heightening the IFN response. So this IRGM can be a very good target for invention of any drugs, which for any kind of inflammatory disorders or viral disorders. More so because in this case we are targeting the host derived factor rather than targeting the pathogen derived factor. So I think this is a plus point in this case. So this is the paper that the work has been published here in IMBO reports. I'm one of the first co-authors here. So thank you. I want to thank my supervisor, Dr. Santosh Chauhan, and Institute of Life Sciences, my collaborators, also my lab mates, and thank you all for the patient hearing. I'm open for any kind of suggestions and questions. Thank you.
Wonderful talk, Parej. It's really great to have you here and listening to this exciting story. A big round of applause for her.
Thank
Now
you.
we move on to the Q&A session. So one question we are getting from Sanchari Gorai. She's asking, is there any relation between IgRM and cancer? She basically means cancer immune escape.
As I've said that IRGM is linked to gastric cancer as far as I know. And till this far, the role of IRGM in inflammation is very well studied. So I am not sure whether it has. I think much studies has been done in IRGM and cancer as such. But like gastric cancer, I'm quite aware of that. But other than that, I don't think that there is any studies regarding that. I don't think that. I mean, I don't know, but I can go and check that.
Okay, great. So I have a question on your last slide where you were talking about IRGM knockout background, where you did see antiviral mechanism, right? So I was wondering if this is so that IRGM depletion results in antiviral state, then what's the purpose of having IRGM?
See, this is like, yeah, I get your thing. This is like having two swords in a same case. Like in one hand, IgM is an anti-inflammatory protein. It is like maintaining the basal homeostasis inside a cell, that whenever a body is getting attacked, it is not letting inflammation to reach to that point where our body gets hampered. But on the other hand, it is like antiviral. So it's like you have to have something in the cost of losing something else.
Right.
Yeah. So like in one hand, it is protecting us, but on the other hand, it is protecting us from deadly viruses.
Yeah.
So it's like two-way sword. So that is very interesting. But it's the same.
It's very
Yeah,
interesting.
it's
So
it's
I was
very
also,
interesting.
yeah. So in the same line, I was just wondering. So in this IRGM depletion background, have you tried rescue experiment to put back a wild type IRGM and see what is the state of viral?
Yeah. I have not. In case of viruses, we are doing it. This is in process till now. We have not done it. But in case of the inflammatory response, type 1 interferon response, we have done it. I have not shown the data here. But when we remove IRGM and then we complement the cells with IRGM, the response goes down. So this we have checked. But in case of viruses, as I said, that we are still doing things. So this is a good idea. We will do it.
Okay. And how much rescue have you found?
Rescue is at a 45 to 50% rescue in case of inflammatory responses. Yeah.
Quite a high amount.
Quite high amount, yes. Especially because we are doing siRNA knockdowns, or we have stable shRNA knockdowns. We are unable to generate CRISPR knockdowns because the cells die.
Okay.
They die. We have tried several times, but complete CRISPR knockdown, it's not possible. So we think that the gene is so much important that maybe not having the gene in case of human. Plus, this is only a single gene, does not have any other isoform. In case of human, I'm talking about, not mice. Mice have like 23 isoforms. So one gene goes away, the other comes and compensates for its function. But in case of humans, a single gene, and it's so important, like you completely just can't remove it at all. So we have been working with like siRNA knockdowns and shRNA cell lines, but the response is equally good as compared to the response that we get in complete knockout mice.
Okay, great. It's really interesting. So maybe
Thank
we
you.
are running out of time. Maybe I cannot
Okay.
take more question now, but it's great to listen to this exciting story you have. With this, I'd request audience
Thank you.
have a virtual big hand for Parej. Thank
Thank
you
you.
for
Thanks a
joining
lot.
us and sharing your.
Thanks for giving me this opportunity. Thank you.
It's our pleasure. With this, I'd move on to our next speaker. She's from Dr. Shweta Chakrabarti. She's from IISc Bengaluru. Currently, IISc Bengaluru. She is now Wellcome Trust DBT Early Career Fellow in at Professor Sandhya S. Vishweswarya Lab. She completed her PhD in life sciences from EPFL, Switzerland, followed by a year-long postdoctoral fellowship. And after that, she joined Professor Vishweswarya's lab back in June 2016. She published an recent article titled Intramacrophage Loss Primed the Innate Immune System via JAK-STAT and Toll Activation, which had been published in Cell Reports recently. She's passionate about understanding the complexity of the human immune system and how this knowledge could be applicable to our daily lives. With this, I'd like to welcome Dr. Shweta Chakrabarti. Stage is yours.
Thank you. I just want to confirm whether my audio is working fine.
Absolutely.
Okay, excellent. So thanks so much to giving me this opportunity to present my work here in this really exciting day of talks in this HerStory of Science. I'm really glad to be part of, this vibrant scientific community and the women scientists of India. I'm really honored to be part of such an event. And today I'll be speaking to you mainly about my work, which I recently published on Drosophila blood cells and how the immune response links to the wounding response. I'll also talk to you briefly about what I'm currently doing and where we're going and where we're thinking of going to next. So the main players of my talk are hemocytes, which you can see here in the center. There's this intestinal tract of Drosophila and these hemocytes, which are in close contact with this tissue. And what I'm interested in understanding is mainly involving the innate immune response. Thanks to the last speaker in explaining a lot about the immune system and DAMPs, so I won't go too much into the details of that. So as you've already probably heard a lot about what DAMPs are, and we know a lot from the mammalian system, but just for me to tell you that DAMPs are these danger associated or damage associated molecular patterns. And what my main question is the mechanisms of how these DAMPs are activated in the context of the fly immune response, so how hemocytes are sensing these DAMPs, and what are the identity of the DAMPs. So we unfortunately don't know a lot in the fly system about that. So my first project was to understand what are these molecules that are triggering the immune response in these hemocytes, and how is this being integrated if there is injury concomitantly with a bacterial infection. So in the case of a systemic immune response, where you have a bacterial infection, say, how do these both pathways get kind of integrated together? And what's the response to the organism health? So my main readouts are usually looking at the entire organism or looking transcriptionally at certain pathways. And the main pathway actually I'll be talking to you about, and which I want to take some time to explain, is JAK-STAT pathway. And why is JAK-STAT pathway interesting for us in this context of wounding is because, as you've already heard from the previous talk, and probably many of you know, JAK-STAT is very important in the context of immunity, but it's also very important in the context of homeostasis in wound repair and in proliferation in animals. And what we are trying to understand in flies is how this pathway contributes to both. And why we use flies as a model system to look at JAK-STAT signaling is because this pathway is much more simplified in the flies. So in flies, you have ligands, which are called unpaired ligands, unpaired 1, 2, and 3. And 3 is the main ligand that's produced in response to damage. And this ligand can then bind to a single receptor called DOMLess, there's a single JAK kinase and a single STAT transcription factor, which then gets activated by this pathway and can translocate to the nucleus and turn on signaling. So what about hemocytes? Are they producing UPD ligands or unpaired ligands after an injury? Well, that's actually what we see. We see that hemocytes, once we isolate RNA from hemocytes from adult flies, we find UPD3 is in fact induced in the injured hemocytes. And we wondered if this response was to purely the injury or maybe to some bacteria or some fungi that sits on the surface of the host. So what we did is we reared these flies in germ-free conditions or azenic conditions, and this AX stands for azenic. And you can see now the induction of UPD3 is as similar as you see in the conventional rare conditions. So this result basically told us that we have an induction of UPD3 to damage alone and not to the bugs which are present in the environment. So we use this UPD3 as a nice readout to look at what's happening in the immune cells after damage and what could be the identity of these dams that turn on this UPD3 signaling. So is this UPD3 signaling important for the flies? What happens if we now block UPD3 production from hemocytes? So this was some of the work I did back in 2016. This work's been published already as well. So what we did was we knocked down individually each of the UPD3 or UPD ligands, UPD1, 2, and 3. And what we found was UPD2 and 3 knockdown from hemocytes. So the genetic system we use in flies is using a GAL4 UA system where we can specifically knock down our gene of interest in the whole organism in our tissue of interest. So this HML stands for hemolectin, which is a driver that will drive the GAL4 UA system only in the hemocytes. And so we knocked down specifically in hemocytes UPD3, and we saw that in fact it was necessary for this the survival. In blue, you can see in the triangles that the flies are dying faster in comparison to the control counterparts. So the next experiment we did was to block STAT. And actually, this is where the crosstalk between the hemocytes and the gut comes in. So this is, again, previous studies that I've done, where we've actually figured out hemocytes signal to the gut and turn on STAT signaling in the gut. And if we block this STAT signaling specifically in the gut, we can recapitulate the phenotype of removing UPD3 ligand from the hemocytes. So using this stem cell driver of the gut, escargot, we knock down STAT specifically in adult flies so that they do not have any problems in development. But what happens is when we knock down STAT in the gut, we see these flies become more susceptible to the wounding. So there seems to be a crosstalk between UPD ligand activating STAT in the gut, which is important for the survival of the flies. And why we thought that this was important was because of this phenotype. So what we found out was in this bar graph, you can see we've tried to quantify basically how well the gut is functioning. And how do we do that? We feed these flies in their food a dye, a blue dye, and you can see that these flies basically either keep the dye in the gut if the gut is functional, so the barrier function is normal. But if the function is dysfunctional, that is that there's a problem with the gut barrier, then you can see the dye leach out and go into circulation, and the whole fly turns blue. And this phenotype has been named by Michael Rera as a Smurf phenotype. So we count the percentage of Smurfs here on the y-axis over time. So in 15 days and 30 days post-wounding. So the CI stands for injury and the SI stands for systemic injury. And you can see in mutant animals, which do not have UPD3 ligand, and also in this case, UPD2 ligand, they start to lose their barrier function even to a normal wound, and of course to a systemic wound as well, in comparison to their wild type counterparts. If you compare the green, light green to the gray, or the dark green to the black, you can see that they are actually becoming Smurf faster than their wild type counterparts. So probably this UPD3 ligand signaling to the gut is required for the proper functioning of the gut. And this was kind of where we were when we started this project. And this was kind of the finding that we reported in this paper. There were other papers as well that showed that there could be a communication between the gut and the hemocyte in the reverse way, that if you damage the gut, the hemocyte can start producing a lot of ligands, such as DPP pathway. And what we found, in addition to other tissues like the liver, like fat body, the gut also had an important role for the organism health. And these hemocytes seem to be at kind of a nexus, is kind of signaling to the fat body, the gut, and different tissues, the muscles, for the normal health of the fly if there's wounding. And so coming back to the question of the DAMPs, and so we wanted to figure out how these hemocytes, we knew that hemocytes were being recruited to the site of the wound in response to H2O2. And this is coming from the work from Will Wood's group and Paul Martin's group, where they've shown that if you knock down one of these oxidases that's responsible for production of H2O2 in flies, hemocytes no longer sense that there is a wound. We wondered if H2O2 was also produced not just in embryos, as they'd shown, but also in adult flies. So we decided to first measure in vivo what the levels of H2O2 are after wounding. And as you can see in these yellow arrows in the panels below, after 30 minutes after wounding, you can see that in fact, there is accumulation of H2O2 within hemocytes and also in the white arrow in the cuticle itself, so where we've wounded. And it seems that this accumulation of H2O2, and so the probe I'm using here is quite specific for H2O2, and this is a boronate-based probe. And so we employed this probe in flies in which we knock down the oxidase, or if we overexpress catalases. So we're basically reducing the number of the molecules of ROS. And what you can see in the panel on the right in the top, if we knock down DUOX, we can no longer see ROS within the hemocytes, and even more so in the cuticle when we knock down or we overexpress USISC, or we knock down the levels of ROS by overexpressing this secreted catalase specifically from hemocytes. So these experiments validated, yes, indeed, there is ROS being produced, or H2O2, in response to damage in adult flies. Now, what happens to their longevity? So the experiment that we've done here is looking at percentage survival. In the red, you have the Duox RNAi flies, and with the overexpression of the catalase in green. And you can see in comparison to the wild type counterparts, these flies are much more susceptible to the wounding. And if we look at UPD levels, if UPD3 is in fact not being induced in these flies, because we knew if we knock down UPD3, these flies are susceptible to wounding. So is there any link between ROS and UPD3? We decided to measure UPD3 levels. So on the graph on the left, you can see that Duox RNAi flies are actually having less of a response of inducing UPD3, and so are the overexpressing catalase flies. So by just knocking down Duox in hemocytes, we seem to impact their response to the wound, and they do not anymore induce UPD3 levels. Now, one experiment that we really wanted to do was to see the sufficiency of Duox. And how we tested that was to overexpress Duox from hemocytes where there was no wound. So is the signal really coming from ROS without any wound? So if we don't wound the flies and just overexpress singly US Duox, and we see what happens to UPD3 production, we see that in fact this was sufficient to induce UPD3 from hemocytes if we just overexpress the protein US Duox. So this shows that not only is it required under wounding conditions, but it's also sufficient for the response of JAK-STAT from the hemocytes if we just express Duox and we have increased ROS levels. So the next thing we wanted to do was look at this intra accumulation. So we saw in the panels before that there was this accumulation of ROS within hemocytes. And the catalase I've been talking about was an extracellular catalase. So we really wanted to remove ROS or dampen the ROS response inside hemocytes. So not touch the ROS in the cuticle and just in the hemocytes. And what we did was overexpress this catalase called catalase A, and that's quite specific for the cytoplasm. And so when we do that, what we found is that similar to the US IRC, we see there's an attenuation of UPD3 in the flies which have been wounded, and the survival of these flies is also affected. Of course, the impact we see in the IRC flies is much more dramatic because we've removed ROS not just from the hemocytes, but also from the cuticle. And here we see that there is in fact a significant impact on their survival. So this, I would say this accumulation of ROS within these hemocytes was something that caught our attention. And we wanted to explore it a bit more. And I'll try to explain you the logic of why that was. So data mining on looking at JAK-STAT pathway mutants. So there are gain of function mutants of JAK-STAT called hopscotch tummel. So hopscotch is the name of the JAK gene and the tummel is the name of the allele. And so what we did was we mined data coming from hemocytes in which these mutants basically show neoplasia. So in mammals as well, in humans, activating JAK mutations lead to blood cancers. So what we found to our surprise was that these gain of function mutants in their hemocytes were showing an upregulation of a water channel. And I'll try to explain to you why that was interesting to us. So this water channel in mammals called aquaporin 1, it's known that it's in fact not just a channel for water, but because of the molecular size of water and H2O2, it can also serve as a channel for H2O2. And PRIP, when we did some molecular modeling, seems to be something similar to aquaporin 1. And so we wondered, is this PRIP upregulation in these JAK-STAT mutants have anything linked to this accumulation of H2O2 within hemocytes? And so we decided to test this. First, we did it in a heterologous system. So we just expressed PRIP channel in HEK cells. And with the same probe that I showed you before, what we found was that there was a faster accumulation of H2O2 within HEK cells when we transfected the cells compared to the untransfected controls. So it seems that indeed this PRIP channel, like aquaporin 1, can in fact help in uptake of H2O2 within cells. But this is HEK cells. What's the physiological significance of PRIP in flies? So first we looked at PRIP localization in hemocytes, and we created a line in which we have basically PRIP, the entire channel, tagged with a superfold GFP, and this is a transgenic fly which is expressing this PRIP channel. And what we found is that it's indeed expressed in hemocytes. And in the second panel, when we added H2O2 at very low concentrations to these hemocytes, what we found was indeed that the PRIP channel was now, not only did we see some sort of an increased expression, but we also saw some sort of a membrane localization of the channel, which was quite interesting to us. So we decided, let's knock this down from hemocytes and not from any other tissue and see what happens to UPD3 induction as a readout of the damage-induced JAK-STAT signaling. And we saw that, in fact, if we remove H2O2 response by just removing PRIP, it looks like, yes, PRIP knockdown, in fact, does lead to an attenuation of UPD induction by these hemocytes. So we implicated a water channel, which was quite new in this JAK-STAT signaling and in the context of wounding, which we were quite excited, and we want to follow this up. We've tried to generate mutants of this using CRISPR strategy, but these seems that these mutants are lethal, but we're trying to follow up a little bit more about the PRIP channel in hemocytes in our further studies. Now coming to the signaling part. Till now, I've only talked to you about how H2O2 basically signals to UPD pathway. Now, what's connecting H2O2 to UPD3 induction? Now, again, if we go back to the literature of migration and how hemocytes home to the site of the wound, we know there's a H2O2 or an oxidative sensitive kinase, which is the Lin homologue called SARC42A. And like Lin in zebrafish, SARC42A also has some cysteine residues, which can get oxidized by H2O2, and then this activates the kinase. So what we did was, in experiments, we knocked down SARC42A only from adult flies in the adult stage and looked at survival to injury and the UPD3 response. And we saw that, in fact, probably the H2O2 response is probably going through, again, the SARC42A kinase, which is responding to H2O2 and activating UPD3 response. Another of targets of the SARC42 kinase is Draper. And Draper is very interesting to us is because it's expressed exclusively in hemocytes and in glial cells. So it's not expressed anywhere else. So we decided to use mutants of this allele. And what we found was that this particular delta 5 allele of Draper was in fact more susceptible to wounding and showed a very dramatic impact on UPD3 induction. So the pathway that we were sort of implicating in induction of UPD3 via this H2O2 molecule was SARC, Draper, and also the Lin target SIC, which is shark, which I'm not showing you the data in because of the shortage of time. So the model that we have so far, and what I've shown you is that you have this ROS burst, you have an accumulation of H2O2, and this is facilitated by this channel, PRIP. You have this pathway that's activated, SARC42A, Draper, shark, that turns on UPD3 molecules. But we're quite focused on JAK-STAT, and we wanted to kind of step back and look at what other pathways could be important in the context of injury and what their role in hemocytes could be. And so to this end, what we did is an RNA-seq experiment with hemocytes. So we collected hemocytes from adult organisms which were wounded. And to our surprise, one of the top category of genes we found were antimicrobial peptides. And in the next slide, I'll get to the details of why we think that is. But just to tell you that we did find UPD3 in our RNA-seq data, And it seems like DUOX is the one which is expressed most highly in hemocytes in comparison to other oxidases. And IRC seems to be, in fact, induced after wounding. So maybe there's some sort of feed forward loop to try and get the ROS levels down so that it doesn't damage the host too much. So talking about these antimicrobial peptides. So these antimicrobial peptides that we found, and in fact, a lot of other targets of a pathway that you must all know about is the toll signaling pathway. So in flies, the toll signaling pathway basically gets activated in response to gram-positive bacteria, fungi. And it's also been shown, in fact, some of these proteases can respond to wounding as well, but we don't know the mechanism there. But what we found was that toll activation in hemocytes occurs because the transcription factor called dorsal, in fact, is nuclear under wounding conditions in hemocytes. So this is the immunostaining that you see on the top. And what we wanted to know was, in fact, just like UPT3, are these antimicrobial peptides being induced in response to a pathogen or the microbiota or just to the wound? And so we employed again the germ-free or azenic flies. And what we found was these antimicrobial peptides were induced similarly in the conventionally reared flies or the azenic flies. And so it looks like the induction of this is via the damage. And is there a link between the ROS signaling and the STOL pathway as well? So we overexpressed, again, DUOX in flies and looked at the antimicrobial peptide cecropin in hemocytes. And we saw that just like UPD3, cecropin induction was also seen after you overexpress DUOX. And was this required for the survival, as we saw in the UPD molecules? And we saw that, in fact, in removing toll seems to have some impact on longevity, but for sure, if we now injure these flies seem to be, in fact, more susceptible to the wound. So this was, again, for us, a new finding that toll is implicated not just in its antibacterial or antifungal role, but also has some role in immunity. And what we're thinking is that maybe these antimicrobial peptides have some role in the wild. And the experiment that we thought was that maybe the STOL pathway is somehow protective for the flies. So the flies get wounded, then if there's subsequently some other pathogen attack, maybe these flies do better. And what the experiment we decided to do to this end was to take naive flies, or UCs being unchallenged flies, clean injured flies, and then infect them 2 days later with a gram-positive bacteria, E. faecalis, and see what happens. And to our surprise, we found that, in fact, flies which have been wounded 2 days previously were, in fact, protected to E. faecalis infection. And we wondered whether this is going through antimicrobial peptides, and there's a heightened antimicrobial peptide to the pathogen after wounding. Of course, there is already an antimicrobial peptide response following wounding, but if we now add in the pathogen, the response is even more heightened. So it seems that this antimicrobial peptide probably could be protecting flies to a pathogen if the flies have been previously wounded. So some sort of immune training is happening to these cells, that if there's a subsequent pathogen attack, these cells do better and help the host deal with this infection. And we wondered if the ROS response was important for this. So we did these experiments in the Draper flies, which don't respond to ROS. And we saw that in these flies, this protection was completely lost. And so was this induction of these antimicrobial peptides. So it seems that, the protection that we have is really via this initial wounding that's taking place, and not because something else that's sticking around and just constantly activating the immune response, and then that's causing the protection. We think it's kind of going through this ROS pathway, priming these cells. These cells already know that there's been a damage, and then when they respond better, but we don't know the mechanism of that. So to that, we want to look at maybe epigenetic mechanisms or something of that, and look at hemocytes and after wounding and see for how long this stays. We've done some time course to look at the resolution of how long this stays. And we've seen after a week, 10 days, this response is lost. So there's some sort of lifetime to it as well. So far, I've told you that hydrogen peroxide from the wound is activating hemocytes. It increases the expression of UPD3. This intracellular ROS is important for their activation. The SARC42 SHARC pathway, Draper pathway, is important for the hemocytes to produce UPD3. And we found out that toll and oxidative stress pathways are induced in response to wounding. Plus, this injury is protective for subsequent infections. So what about the mechanism? So as I said, we're interested in looking at epigenetic mechanisms, and we're also interested in looking at what other pathogens could be. Maybe the fly is not just protected against gram-positive bacteria, but also against fungi, maybe gram-negative. So maybe the mechanism of antimicrobial peptide could be some sort of protection rather than just pathogen killing. So that's something that we're trying to look at right now. So in my acknowledgements, I'd like to thank Professor Sandhya and her lab, and of course my mentor Irene, who's been very helpful in all the guidance she's given in this project, as she's one of the mentors in the ECF, and of course India Alliance for the funding. And thanks for your time, and I'd be open to take any questions. Thanks.
Thank you, Shweta, for sharing your exciting story. Very extensive study you have done using this Drosophila model system. So it's time for Q&A session. So we got some question. So this is from Imroz Khan. He's asking.
Yes. Hi, Imroz. Yes.
He's asking. He's asking, is it because E. faecalis itself potentially works via ROS activation in flies after infection? Prior ROS activation prepares the flies for better survival against EF infection. Have you tested the same for other pathogens also?
No, so as I said in my talk, unfortunately, we haven't tested other pathogens. That's something I'm really interested in testing. I want to test other pathogens that flies would possibly see in the wild. And I want to look at different panels of not just bacteria, but fungi as well. And so that's something I really want to see how this protection mechanism is. This ROS mediated, like he's saying, or, are these antimicrobial peptides? I mean, there's a lot of talk these days, at least in the gut, that these antimicrobial peptides may have some other role other than just their pathogen killing, these cationic molecules. So, I mean, maybe our system has some ways of looking at that. And so we want to look at that in more detail. So I think it's a very important question. And we are doing those experiments now. We'd like to test how this, this training is working specifically. Is it specific to E. faecalis? Or is it more, prevalent for other pathogens? Thanks for that question. Yeah.
Thank you. Another question we got from Mitali Singh. She's asking how long the protection lasts.
Again, I didn't show that in my experiments, but as I said, we've tested that the protection actually lasts for about a week, 10 days. So after which, when I start to do these experiments, I no longer see this sort of a protection. So there's some sort of life to it as well. And I don't know if it has to do with the blood cells, because there's a certain number of blood cells in the adult fly. There's little hematopoiesis. So then maybe the population that's protective sort of dies out, or they are not functional anymore, something of that sort. I don't know what the reasons for that is. So we also want to look in single cell RNA-seq experiments to see if there's a kind of subpopulation of cells, or is it a more ubiquitous thing of all hemocytes? Are all hemocytes now behaving this way? Or is it like, because my assay is a whole, all the hemocytes are taken them. I haven't resolved into if there's a subpopulation, the ones which respond, go to the wound. And so those are kind of things I really want to see in the future on how, these hemocytes respond. And of course, in the lifespan of the fly, how they change and how they could contribute to the other physiology. So the gut part I didn't talk about, and that's what I'm working on. So are these cells now hanging out in different tissues or hanging out in different parts of the body and preparing the host for other, threats other than just a systemic immune response? Yeah. Thank you.
Really exciting. Would you mind if I take another question?
No problem. Please do.
Please, thank you. This is from Arun Prakash. He's asking, is the protection mainly due to E. faecalis clearance from AMPs?
Yeah, so the clearance experiment, I mean, it looks like there is definitely some killing of the bug that's happening, but I will need to look into this more carefully. So the CF experiments we've done are sort of preliminary. So that's what I want to know, if these antimicrobial peptides are really for clearance, or is there some sort of, tolerance that's happening? So to figure that out, I need to do a little bit more experiments, and we plan to do more experiments in the future for that. I hope I'll be able to answer this with my next presentation. Yeah.
Great. I have a question. This is on whether this UPT3 have any formula.
Yes, so it's IL-6-like. I'm sorry, in my introduction, I didn't explain that well. So UPD3 is IL-6-like. UPD2, in fact, is leptin-like. So they're quite different to each other. They're sitting right next to each other on the chromosome, but one seems to be more like a metabolic kind of a ligand, while the other one is more immune ligand. So yes, that's what their homologs are in mammals. Yes.
Great, exciting.
Yeah.
If there is no more question, then with this, I'd like to conclude this session. Thanks again. Thanks a lot for sharing your exciting story with us and joining us here in this session. It's our absolute pleasure. Again, a big hand to Dr. Shweta. With this, we'll conclude this session. And we'll again come back sharp at 7 with the session, our first keynote session, where is this visible? Our first keynote session, where Professor Nahid Ali from CSIR Indian Institute of Chemical Biology, Kolkata, will talk. So don't go away and stay tuned. We'll be just back sharp at 7. Thank you for listening to us. Good evening. আর কেউ নেই না? না,
চলো। আচ্ছা, এটা বন্ধ করি তাহলে।
হ্যাঁ করে দাও।
লাইভটা বন্ধ করছি, এটাও বন্ধ করছি। লাইভ স্ট্রিম তো